Hello, I'm Francesca. I'm a first year PhD student in the Saunders Lab at the John Innes Centre, and today I'm going to tell you about the important role the chloroplasts play in the plant immune response and how I'm aiming to understand how the rust fungi in particular manipulate the weak chloroplasts and photosynthetic processes during infection. So, through many studies over the years, we know that plants have a complex immune system to detect pathogen invasion, mount a defense response, and ultimately prevent infection. And plants can detect pathogens at the cell surface through the recognition of highly conserved pathogen molecules, known as PAMPs, which might be, for example, chitin on the fungal cell wall, or flagellin, which is a component of bacterial flagella. But pathogens have evolved specialized proteins called effectors, which are secreted into the apoplast or into the host cell cytoplasm. And these proteins act to manipulate host cellular processes, including, importantly, to suppress that first layer of recognition. However, plants have evolved resistance proteins to detect these intracellular effector proteins, either directly in a gene-for-gene interaction or indirectly through their biochemical activity. And so through the recognition of either PAMPs or effectors at either the cell surface or within the cytoplasm, signals are transduced to the nucleus, resulting in global transcriptional reprogramming and the initiation of strategies to prevent pathogen growth, such as strengthening of the cell wall with callos, or in the case of a strong immune response triggered by effector recognition, a localized cell death known as the hypersensitive response. And the chloroplasts have a key role in initiating and coordinating the plant immune response. Firstly, not only is immunity an intensely energy demanding process, and that energy comes from the photosynthetic processes of the chloroplasts, but the chloroplasts are also the site for the synthesis and release of a number of key pro-defense molecules, such as reactive oxygen species, and importantly for immunity against biotrophic pathogens such as rust fungi, salicylic acid. And pathogens have evolved effectors to target and manipulate chloroplast processes to interfere with the plant's immune response. An example of this is the Eustelago maydis or smut fungus effector protein CMU1, which manipulates salicylic acid biosynthesis to metabolically prime cells for infection. And the hop effectors, secreted by the bacterial pathogen Pseudomonas syringae, which target reactive oxygen species production and salicylic acid biosynthesis to interfere with immune signaling. And so there is a large and growing body of evidence which supports the varied and important role of the chloroplasts in enabling a robust immune response against pathogens. And there is evidence from our field as well that this is also true for the wheat-PST interaction, with two exciting studies coming out last year which demonstrated this. Firstly, we have the further characterization of the mechanism of YR36-mediated immunity. So YR36 is a high-temperature adult plant resistance gene and confers resistance to a number of PST isolates. And in this study, it was shown that the protein which YR36 encodes for phosphorylates the photosynthetic protein PSBO, which leads to its degradation. Loss of this protein from the photosystem II complex leads to inhibition of photosynthetic processes and induces chlorosis, preventing PST growth and thus providing the plant with resistance to PST. And then another study focusing on the targeting of the chloroplasts by PST revealed that the effector PST 12806 targets an iron sulfur protein in the thylakoid membrane. And this prevents the buildup of reactive oxygen species and blocks that cell death response, enabling PST to colonize the plant. And so these studies have begun to reveal the dual complexities of chloroplastic manipulation by PST, and also the importance of chloroplastic processes in providing immunity against PST. And so the question of my PhD is, how can we further our understanding of how and why the weak chloroplasts and photosynthetic processes are being manipulated during infection with rust fungi? And to begin to answer this question, a previous PhD student in the lab carried out RNA sequencing of wheat tissue infected with PST. And this revealed that the expression of some nuclear-encoded chloroplast genes are being modulated over the course of infection. So if we take a look at this example of a nuclear-encoded chloroplast gene, we can see that the expression of this gene is severely downregulated at 1 day post-inoculation. And this is followed by an increase in expression at 3 days post-inoculation. And this kind of early expression pattern is also seen in other pathosystems. And we know that in general, photosynthetic processes are inhibited early during infection. However, interestingly, in the resistant interaction, there is a consistently higher expression of these chloroplast-associated genes than in the susceptible varieties in the later stages of infection. And we hypothesize that the higher expression of these genes in the resistant variety is an indication that these genes and the proteins they encode are important determinants of susceptibility to PST. And over the past few months, I've been validating this data and have begun to investigate the function of some of these genes. So my overall strategy is to use a reverse genetics approach to identify chloroplastic genes of interest and functionally characterize their role in the wheat response to rust fungi. Firstly, using the RNA sequencing data, I have identified a number of nuclear-encoded chloroplast genes of interest which follow this general pattern of expression. I have utilized the tilling population and obtained single loss-of-function wheat mutants, and I'm currently generating double loss-of-function mutants. And these mutants will be screened for changes in the level of susceptibility to wheat rust fungi. And those mutants, and therefore those genes which show to be involved in the wheat response to rust fungi, will be taken forward for functional characterization. And one of these genes is already through the pipeline for functional characterization. So a chloroplast-associated gene was selected using the RNA sequencing data. Loss-of-function tilling mutants were obtained, and these mutants were screened with PST. And so here you can see the heavily infected wild-type plants on the left, and the loss-of-function mutants with a reduced susceptibility to PST on the right. And we are currently generating double loss-of-function mutants, which again will be screened, and then we will begin to functionally characterize this gene. And so this is a really nice example of how powerful the RNA sequencing time course data can be for identifying genes of interest to enable us to elucidate mechanisms of chloroplastic alterations during infection. And so overall, across the next 3 years, I hope to contribute to this field by increasing our understanding of how chloroplastic processes are being altered by wheat rust fungi during infection. And I'm excited to share more with you over the coming years as my project progresses. So I would like to thank my main supervisors, Diane Saunders and Nick Talbot, and my postdoc supervisors, Pilar and Ayako. And I would like to thank the whole of the Saunders Lab, who are a really great team to be part of and have been incredibly supportive, especially during lockdown. Thank you to BGRI for giving me the opportunity to present today. And thank you for listening. I'll be happy to answer any questions during the Q&A.